गुड आफ्टरनून अंजलि मैडम।
गुड आफ्टरनून सर।
अरे वाह, गीता मैडम भी हैं।
गुड आफ्टरनून मैम।
Good afternoon, ma'am. Thank you for understanding that it was a very important meeting. इसलिए मैंने थोड़ा सा शेड्यूल करवाया.
Thanks to you, ma'am, for accepting our invitation.
Anytime, ma'am. Anytime.
Good afternoon, sir. Good afternoon, ma'am. Actually, I was mute that time.
आपको किसने चुप कर दिया मैडम?
गीता मैम ने.
अच्छा।
मैम मैंने तो प्लेटफॉर्म दिया आपको बोलने के लिए, ऐसे मत बोलो।
Ma'am, shall we start?
हाँ, yes ma'am, yes please.
So, a very good afternoon to all the respected dignitaries, our Sailesh sir, our esteemed speaker, Dr. Sufla Gupta ma'am, faculty members, researchers, students, and participants joining us today. I, Dr. Anjali Sharma, feels privileged to welcome you all to this national seminar on From Molecules to Mechanism, Multi-omic in Plant Research, organized by IPGA Women Forum, Haryana State Branch, in collaboration with Guru Gobind Singh College of Pharmacy, Yamunanagar, Amar Shahid Baba Ajit Singh Jujhar Singh Memorial College of Pharmacy, Bela, Ropar, and EduSpec. Today's seminar aims to explore the fascinating journey from molecular insight to biological mechanism, highlighting how multi-omics technologies are evolved in plant research and modern science. For today's session, we are privileged to have with us Dr. Sufla Gupta ma'am, Scientist F, Plant Science and Agrotechnology Division, CSIR, Triple IM, Jammu. I really welcome you, ma'am. And I also welcome Sailesh sir from Bela College. I welcome you, sir. And before we proceed further, my sincere gratitude to Dr. Manju Singh ma'am, the chairperson of IPGA Women Forum, Dr. Vandana Garg ma'am, co-chairperson IPGA Women Forum, Dr. Geeta Deswal ma'am, the coordinator IPGA Women Forum, Haryana State Branch, and Dr. Neelam Sharma ma'am, coordinator IPGA Women Forum, Punjab State Branch. To formally commence with the academic gathering, I would like to invite Dr. Geeta Despal ma'am, the organizing secretary of the seminar, to deliver the welcome address. Geeta ma'am, please.
Thank you, Dr. Anjali. Good afternoon to all respected dignitaries, our esteemed speaker, Dr. Sufla Gupta, Dr. Sailesh Sharma, Director, ASBUS JSM College of Pharmacy, Bela, and faculty members, researchers, and dear students. It is my great pleasure and privilege to extend a warm welcome to all of you to this national seminar on From Molecule to Mechanisms, Multi-omics in Plant Research. This seminar is being organized by IPGA Haryana State Branch in collaboration with IPGA Women Forum Punjab State Branch and in collaboration with Guru Gobind Singh College of Pharmacy, Yamunanagar, ASBUS JSM College of Pharmacy, Bela, and EduSpread. We are truly delighted to see such a enthusiastic participation from academicians, researchers, and students who share a common interest in advancing scientific knowledge. We feel especially honored to have with us Dr. Sufla Gupta, Principal Scientist from CSIR Triple M Jammu, whose expertise and research contributions in this field will certainly enrich our understanding. We also express our sincere gratitude to Dr. Zabir Ahmed, Director of CSIR Triple IM Jammu, for his kind support and encouragement in making this academic interaction possible. I would also like to extend my heartfelt thanks to Dr. Manju Singh, Dr. Vandana Garg, and Dr. Neelam Sharma for their constant guidance and valuable support in organizing this seminar. And with these words, I once again extend a warm welcome to all of you and wish everyone a very insightful and engaging, productive session ahead.
Thank you so much, ma'am, for your warm and inspiring words of welcome. So now it's time to move toward our technical session. And it's my great honor to introduce our distinguished speaker of the day, Dr. Sufla Gupta, ma'am. Dr. Sufla B. Gupta is scientist in the Plant Science and Agrotechnology Division at CSIR IIM Jammu. Her research expertise encompasses plant molecular biology, functional genomics, DNA fingerprinting, barcoding, tissue culture, and plant proteomics with a great emphasis on understanding and enhancing secondary metabolite biosynthesis in the medicinal and aromatic plants. She is deeply involved in translational research under the CSIR Aroma mission, where she serves as a nodal scientist coordinating with farmers for the production of quality planting material, scientific cultivation practices, and value addition of aromatic oils aimed at sustainable rural development. Her work focuses on large-scale cultivation of aromatic crops such as lavender and rosemary across Jammu and Kashmir, Uttarakhand, and northeastern India. Notably, lavender cultivation for farmers in the small village of Doda resulted in purple revolution in Jammu region, fostering agri startups, generating employment, and significantly strengthening the local rural economy. The collective efforts by the farmer and CSIR team was recognized with the Rashtriya Vigyan Team Award 2025 from the Government of India. And it's very wonderful, ma'am, and we are privileged to have you with us. Dr. Sufla B. Gupta, ma'am, has over 25 years of research experience in medicinal plant biology with significant contribution to crops such as opium poppy, ginger, stevia, and glycyrrhiza. Dr. Gupta has successfully completed more than 11 major research projects and continues to lead or participate in several national scientific initiatives. She has authored over 80 international peer-reviewed publications and delivered numerous invited lectures at national and international forums. So, we are truly delighted to have her with us today to share her knowledge on the topic from molecules to mechanism, multi-omics in plant research. I request ma'am to please kindly deliver her lecture. Ma'am, the virtual stage is all yours. Ma'am, please.
Thank you so much, ma'am, for such a big introduction. I should
My
have.
pleasure, ma'am.
So no, while listening to you, I was thinking I have done something which starts from research. Am I audible? Am I audible?
Yes, ma'am. You are audible.
So listening to my own introduction, I was thinking I started my journey with the lab research, and I'm going to end this whole journey, scientific endeavor in societal application of science, which is such a great satisfaction when you do something in the lab, and then you can transform or translate that work and take it to the farmers. So basically, I'm a plant biologist and love plants. So plants, whether they are in the lab or in the fields, it makes the same passion to me. So today I don't know if we have students of PG and UG both or only PG. Only PG.
Both, ma'am.
Both, ma'am, both.
So please, this is my request to the students that I will be, if it is very difficult for you to understand, stop me there. I will be I will try to explain it in a very simple form again. Because doing research, then you tend to forget what PG and UG textbooks generally tell you. So please stop me, and then we can move ahead. It will be an interactive session, and I will be basically showing all the research that my student had done in the lab. I'll be concentrating instead of plants, many plants. I'll be concentrating on glycyrrhiza glandula. So what we have done, I'm going to share my slide now. What we have done in now. Can everybody see this? Is it my slide being shown?
Yes,
Yes.
ma'am.
And are they changing?
Yes, ma'am.
Fine. So from molecules to mechanism, this is like when we start something, we tend to first focus on its morphology, then chemistry, and then back inside what is the science behind the whole thing. So this will be integrating many different aspects of a plant, from morphology to proteomics to genomics, and then back to molecule. So how it is affecting a plant to withstand the environmental conditions, whether it is stress or a normal ones. And how omics or AI in today's world is helping to integrate all these omics. So what we see generally is a very small product of what is going on inside the plant. Now that is being catered. So generally earlier the science was mainly focused on single molecule or single protein or single gene. Now this is the time where the plant as a whole is being studied in terms of high throughput technologies to generate a large amount of biological data, and this data is further through AI and through bioinformatics. It is being converted into different specific level of interaction, and then that knowledge is being used for using the plant, understanding the plant secondary metabolite, and then translation of that secondary metabolite. Now the whole foundation of the whole thing is again the DNA. DNA is the starting point from where we try to understand how plant is going to behave in a certain conditions. And in the omics, DNA is being studied through genomics. So genomics is the genetic potential and variation in a plant. Then what genome gives you DNA, cDNA, protein, and it is then transcribed into regulatory responses through genes, which is transcriptomics, then proteomics. Proteomics, how the protein or the enzyme function inside the plant. Then this is again a series of multi-step reactions, and then metabolomics. What is being formed in these reactions comes under metabolomics, and there is also epigenomics, something which is not being governed by the ATGC, the alphabets of the genetic makeup. It is also being governed by how expression of gene is being controlled inside the plant, and then this whole system comes under the system biology framework. All these 4 or 5 omics are being studied nowadays sequentially, simultaneously, and interactively to understand how plant behaves. So it starts from RNA sequence. So it starts from RNA, forms protein, that RNA differentially expressed proteins are identified. Then what these proteins actually do inside the plant, that is being studied, and what these proteins or enzymes are converting into, what are the pathways where they are working, that is being studied. And these three, all of them integrate to form what is the interpretation of metabolites, genes, differentially expressed genes inside the plant. Now, so when we do proteomics or genomics, a lot of data is generated, and not all of them, the whole of data is of use to us. So, and also these data are of several features. For example, if you are going to take, now, how do we take the data? So when there's a plant, you want to take the data of the plant. Say, suppose one bioactive compound is being produced in the plant. Here it is glycyrrhizin, which is being produced by glycyrrhiza glabra. And this is a triterpenoid which has anti-cancer activity. Now, if this anti-cancer, how this anti-cancer activity can be enhanced, that is being studied under research. So if the plant is being kept under stressed condition, how the whole genome of the plant or the transcriptome of the plant is being altered, that is being studied through transcriptomics. Now, once this is done, we know, and then you compare this with the control plant. So you are keeping your plant, so for example, under cold conditions, and then another plant keeping it in a normal condition, and then you do transcriptomic analysis of plant A, which is under cold condition, and also plant B, which is under normal condition. And then huge amount of data is generated through transcriptomics. Then you start differentiating the two data. I mean, start comparing them and differentiating what are the genes which are being, because since the plant is same, the transcriptome will be same. The only alternation will be when you are keeping it in different conditions. So all those genes which are being upregulated or downregulated, that is, their expression is higher or lower under cold stress, will be altered. Only those genes or those metabolic pathways or those metabolites will be altered. So you compare the control plant with the treated plant and identify the set of genes, the subset of genes which is changed. They can be highly expression of those genes can be higher or they can be suppressed, lower. Now these genes are very important, and these genes are called differentially expressed genes. Let us not discuss this. But there are certain, very subtle things which needs to be identified when you do omics data. It does not follow what you want it to give the result. It will only be possible for the data to be factual if it is statistically correct. For example, if you are keeping one plant under one condition, the other plant under a different condition for one experiment, it is not necessary that the other experiment will also give you the same result. So the result will vary depending on the situation you are keeping, the age of the plant, what is the temperature also. If it is 4 degrees, it will give you some other subset of expressed genes. If it is 7 degrees, it will give you some other subset of genes which are expressed or downregulated. So omics does not follow exactly what you want it to give you every time that you are doing the experiment. And the results become descriptive, not predictive. So your data is always based on what is the initial condition. What is the age of the plant? What is the conditions where you have kept in? What are the software that you have used? So it will vary with all these variations. And then you cannot compare your data with the published data. Understood? So it is not possible that you say if your result are coming to be A, B, C genes, and the published result says it is D, E, F. It is justifiable. It need not repeat the already published data. And because the AI has this tool which has high dimensionality, it can cover several other factors into account when you are doing the software analysis. And then how do you integrate multi-omic layer? That is also important when you do the data analysis. Okay. Now, when you start your research, the first and foremost is what is that you want to understand. For example, if you want to understand what are the genes which are giving you higher bioactives under drought conditions, that initial data you have to be very clear that it will give you enhanced bioactive. That is what I meant by this is that you have to have the preliminary data of chemistry, of chemical analysis, which is showing you that yes, if you are giving this plant drought conditions, it will give you a metabolite B in higher amount. That has to be with you before you start an experiment. Because otherwise, just by thinking that you are giving stress to a plant and it will give you the results, that is not possible. So a hypothesis has to be there that yes, I have given my plant these conditions and it has given me glycyrrhizin in a higher amount as compared to the control plant. So you will move ahead with that hypothesis and then start proving it in your further experimentation. So prediction and classification. So what are the, so no, when you are giving this transcriptional analysis, the data comes in GBs. I mean, it comes in gigabytes. Such huge data it comes. So you have to understand out of this thousand and thousand of genes, which is the area that you are targeting. Here, your research will help you that yes, for terpenoid biosynthesis, for glycyrrhizin biosynthesis, terpenoid biosynthetic pathway is the one which is getting affected. So, and for phenylpropanoid, for your valeriana, valerianic acid, phenylpropanoid pathway is getting affected. So that pathway, you have to be very clear in your mind out of this thousand and thousand genes, which pathway you are targeting. So prediction and classification is essential when you do your work. Then relationship modeling comes with AI. You have to understand that the data that you have got, now how are you going to visualize that data and present it through modeling? And then this comes under data learning. This is all, without this also we can do our experiments, but this is all to enhance the precision and also the visualization of the data in a smaller time. Now, this is for the UGs and PGs. Try and understand this. For fundamental discoveries, omics approaches are like molecular signatures. So, through omics, you know what are the set of genes which are being altered, not a single gene. So it is a molecular signature. So you will be targeting this set of genes when you do further analysis. I'll be showing that in my experiments when I move ahead. Then precision diagnostics is again one important thing, that once you know that these set of genes are being changed, you have to validate that data in the lab to understand, yes, this is the set of gene which is actually being altered and not no other ones. Then personalized medicine is something where what is that you want to target, and then you engineer that pathway to produce the medicine that you want to, the bioactives that you want to produce. And translation research, then this is the data that you have, then you go back to your lab and then you do bench analysis. That is, you set up, you know that these are the results that has come, and now you want to validate them in the lab through different molecular biology tool, that yes, actually this is not only not the statistical numbers, they are actually being upregulated or downregulated in the plant. Then so this is for all those people who want to go into research, and then how to initiate one research. Once you are into the lab, you have to understand there are n number of plants, but people are working on almost all of them. So you have to identify the gap areas in that particular plant where the work has not been done, or where your work research work will help to augment the already existing knowledge. So for this, I am very sure nowadays people are on LinkedIn and all, and they know what are the tools which help you in doing this. So LinkedIn is a very good app where you can sit down and take out all those information through AI, which will help you in understanding the research part that is being done and what is the white space or the uncovered areas which can be further researched. So this is one such example. When we started our UGT is UDP glucosyl transferases. These are the enzymes which convert metabolite into its glycosidic form. So for example, it is diosgenin, so glycosides of diosgenin, like that. So under these conditions, when we did our research analysis, we could see that there are only paper number 21 and paper number 17 and 23, which are being highly cited by people. That indirectly means that these are the ones which have information, fundamental basic information, which are required for other papers to move forward. So this is the information seen, and that too in January 2021, 21 papers were published, and 21 paper was the one which was being cited. 33 papers were published, 38, and 21 paper is the one. Paper number 21 is the one which is highly cited. After that, January 24, although papers were published, but they did not give you information enough to take the reference from and move ahead. So this kind of data will tell you what are the papers that has been published earlier which are important for your research. And when you see this paper number 21, and this paper was like a very breakthrough in glucosyl transferase activity. These are the enzymes, as I said, they add a glucose molecule to a substrate and make them glucosidic. So why do a plant need a glycosidic molecule? Because these molecules are soluble molecules, and solubilization of a metabolite helps them to store them properly. And also they are their bioactivity wise, the bioactivity is enhanced, and it is easily absorbed by the human body. So glycosides are very important for pharmacy as well. Now, so this particular paper showed for the first time C glycosylation. So normally O glycosylation is reported. Not many papers have reported O glycosylation at the O position of the molecule. This paper also reported for the first time that a molecule's C position is also being glycosylated by these enzymes. And this actually enhanced the application of the UGTs in plant. Now, yes. Now, this is looking little complicated kind of a thing, but very easy to understand that when you do whole genome or transcriptome analysis, data, as I said, is very huge. Such huge data cannot be given in Excel sheets. It is normally visualized. And through the visualization only, you can understand what is important and what has to be taken out. Because you cannot read each and every paragraph of the report. So here, it is actually telling you the Glycyrrhiza uralensis species, how it is different from other known species which are there in Fabaceae family. And if it is there, which is the closest one to this particular Glycyrrhiza species. Only this little information, but this little information is highly precise and 100% correct. So now you know this Glycyrrhiza. If you want to move ahead in your molecular biology, which is the plant you should be talking to? Which is the plant which is most closely related to your plant? And if anything which is being published in plant A, which is closely related to your plant, can be extrapolated to a glycyrrhiza as well. So this makes the whole study easier. Now, this is again, so this particular transcriptome analysis was, as I said, not many information was there, but my concern was only UGTs, UDP glucosyl transferases. So out of that GB data, multifold GB data, I only concentrated, or my lab only concentrated on the UGTs, UDP glucosyl transferases that are present in glycyrrhiza. Why? Because glycyrrhiza has not many flavonoids and terpenoids, and these are all, majority of them are glycosylated. So there must be n number of UGTs present in them. First point. Second point, no such study was done earlier. So for doing a research, these two are the important criteria for moving forward. One, the study is useful to understand a mechanism, and second, it has not been done earlier, because then you will be adding to what is existing in the information. Now these are all AI tools, data visualization. Since lot of data was there, these tools were used to visualize the data in a very easy way to understand. For example, here you can see that, I know this is not very clear writing, but the red one, it says that cytoplasmic inheritance. So it shows that most of your genes were, most of the UGTs were present in the cytoplasm. So this is the area where one has to concentrate. The red color meaning that 40 or above, and the size means 20 times. So these were the set of genes which were important, which was important for us. Secondly, this data shows cellular localization as radical heat map. So this is one, meaning all those orange lines are the ones where the localization of UDP glucosyl transferases is highest. And these are the UGTs, the numbers which are shown in the outer side. And this is the one which is highly present in, this is plastid. These are the organelles where it is present. So this is again visualization of the data, that which gene is present in which part of the organization or the organ. Now, the gene was, so this particular research, in this particular research, we were able to identify 83 such genes from the plant. And how do you identify? The genes have a signature sequence, a consensus motif, which help you to mine the gene from a number of genes. So there are tools which tell you, when you put the data, the tool will tell you that this particular domain is present in this gene, and so it belongs to this class. So this comprehensive domain analysis was done to understand the presence of glucosyl transferase motif in the genes. Okay, this is all molecular. So let us come to this. So the idea was to understand. I have kept one of my plant in control conditions, the other one under normal condition, and was trying to find out what are the genes which were present in the cold conditions, highly expressed in the cold conditions. So as you can see, these 3-4 red lines are the ones where log2 value is very, very high, meaning that these are the genes where I should be concentrating when I do my research. So UGTs or UDP glucosyl transferases can be involved in n number of these processes, metabolic processes. But the ones which are highly expressed under cold conditions will normally be involved in a similar kind of processes which are interlinked. So out of 83 genes, this data helped me to concentrate on 5 genes. So now you can understand the use of AI in your transcriptome, that instead of analysis of all the 83 genes, you are now doing only analysis for these 5 or 6 genes which are highly expressed under cold conditions. You understood? Then how do you do that? Okay, your data told you, yes, they are highly expressed, but then they need to be confirmed in the lab. Now, how do you do that? You do real-time analysis. Real-time analysis is like the plant which is kept under cold condition and the plant which is kept under normal condition. That plant you take out RNA, then you make cDNA out of it, and then using all those 5-6 genes in primers, you make primers of them, and these primers you do real-time analysis and then PCR, and you come to know what is the difference between your control plant and then your cold color conditions. So the ones here, the red colors are the genes. These are the genes. This is heat map, and the gene C. This red color shows that this particular gene is being highly expressed in shoot tissues under pathogen. That is, so you have a plant. Okay. So before we went to cold conditions, as I said, you have to have a preliminary data that why you want to go for cold. So the girl who was doing her PhD, she tried all different stresses and then came under one condition, cold conditions where maximum genes are being expressed. Meaning that cold condition is the condition where UGT is being involved in a very large amount. So it's not taking drought because only one gene is there, not pathogen, only one gene is highly expressed. Only under cold conditions, not many genes were expressed. So this tissue and that too under shoot tissues. So this tissue was taken and cold conditions were given to the plant to understand the role of all these 5 genes in cold conditions. So this narrowed your research to a point where you can do the research. You cannot do research with all the 83 genes. Then you have to concentrate to only ones which are important. Now, how do you do it? Then you clone all these genes. All 5 genes were full length cloned. Cloning is done through PCR. You make primers. It is easy to make primers when you have transcriptomic data because you have all the sequences with you. So you make 5' primer and you make 3' primers and then amplify the gene in PCR. And you have the genes of required size amplified. Now once this is done, these are then used for cloning them or subcloning them in expression vector. So why do you need to clone them in expression vector? Because you want to understand if this gene is being expressed and then protein is formed and what this protein is doing to your plant. If you are not expressing your gene with the expression promoter, then your gene will not tell you what exactly it is meant for. So just cloning will not help. You have to express this gene in a plant. And not only gene, you also have to have its upstream region, the promoter region, to understand what are the motifs or what are the sequences which are present in the upstream region that are important for cold induction. Okay, so now let us skip this. So once you clone this into the vector which is expressed in plant or bacteria, then you know what is the protein which is formed. In this particular case, we express this gene, these 2 genes out of the 5 into the bacterial system. And so when you express them in the bacterial system, you again confirm that they have got integrated into the plasmid that you have added into the bacteria. So once this is done, then you give them induction. This is this protein slide which tells you that when you extract the protein from the bacteria, your gene is being produced in higher amount in the bacteria, which was not there in the control condition. This is your control protein. This is protein induced with 0.2 micromolar of IPTG. And then you start increasing to 0.4, 0.6, 0.8. And then you try to find out how this gene is maximally expressed in which concentration of IPTG. This is your marker. So this marker is required because you want to know what is the molecular size of your molecular weight of your protein. Now you know your protein is being induced in the bacteria, meaning it is expressed. Now confidently you can move ahead that this must be a functional protein. So what do you do to take that protein out? Then you do column chromatography to take out only that gene here, 54 kDa, which is important for you. Yeah. This one, this is your molecular weight marker. And this is the gene. Out of so many of them, you took out through column chromatography only the gene which was expressed in your bacteria from outside. Now you have a protein, not gene, sorry, protein. Now you have a protein which is functional, and then you can use this for confirming that it is actually doing glycosylation. As simple as this. So it starts from induction, it starts from cloning, then expression, then induction, and then it goes to your catalysis, biocatalyst. Now, also it is important to understand where this gene is localized. So the gene localization is important because the functional activity of a protein is not at the place where it is being transcribed. For after translation, it moves, and the site of action is the place where it is actually being glycosylated. So you try and understand where exactly it is being transcribed and where it is translated. Now, so now you have the gene, and then you do in vitro enzyme assays, meaning you have substrate, you have glucose, substrate that can be glycosylated. Not all substrates are glycosylated. So substrate that can be glycosylated, you have glucose to help them glycosylation, you have buffer, and then you have that protein that you have expressed. So this epitope of yours will have all these components, and then you will do incubation to keep it into for it to work and glycosylate. And once it is done, say for example, glycosylation does not take more than 30 minutes. It is a faster process if everything is pure inside your epitope. So say after 30 minutes, then you take it out and you do LCMS analysis to understand if the molecule is being added to your substrate or not. This is very interesting to understand. Always I have seen these, all those people who have worked on plant biology are not very easily moved to analytical chemistry. But it is when we are doing our research, it is mandatory to understand how a transition takes place from plant biology to a chemistry inside the plant. So this is like, suppose here the substrate is camphorol. Substrate is camphorol. It has a molecular weight, and if 162 glucose is 162, if 162 is added to this molecular weight, you know your glycosylation has been done. That is a simple calculation, but that peak has to come. And there are several times where glycosylation is not being done at one point. It can glycosylate different functional groups at different points. So it cannot be monoglycosylation. It can be a diglycosylate or a multifold glycosylation. So that has to be understood, and that has to be proved through chemistry that yes, it is being glycosylated. So you take the substrate and see the glycosylation, and then you further break it down and see if when you remove this glycosylated molecule, you go back to your original substrate. So this is precursor ion scanning, and then you confirm that it is glycosylated. This is important because there are no standards available for glycosylated products, very, very few. And since there are n number of flavonoids and terpenoids, not all standards or reference markers are available. So this is how you prove your point that yes, it is being glycosylated, and now it has become a glycol. That's all. Is it 45 minutes over? No, okay. So this is my research lab. These Bhavna and Shenawaz, these two have done this glycosylation experiments, and
हाँ जी
that's all.
हाँ जी
Yes, sir.
जी।
Uh,
ये
ma'am,
कांच वाले हैं, इनने कांच की नाप लेनी है कॉलेज के अंदर, मिरर की नाप लेनी है। मैं प्रिंसिपल बोल रहा हूँ फार्मेसी में।
there is one question from inform student.
Yes, ma'am.
The question is, what are the main challenges that arise while using omics data for discovering drug from plants?
Main challenges.
Yes, ma'am. They're asking about the challenges.
Okay. So the first and foremost is because these metabolites are a huge. No, it is a very plants are very big repository of metabolites. So you don't know what is the structure, and then you don't know there are several metabolites which are not known to the literature. So you won't be able to give that data to your AI platform. So the AI works only on those data where only on those clues or cues where it has the initial information. If that information is not available with the plant, the software, it will not be able to tell you the exact properties, the structure, and also it can maximum it will be able to tell you the molecular weight and all. But other things like what is the NMR structure data of it, that one has to isolate it and do it. So AI can only help you in understanding yes, it is closer to this molecule. But actually it is closer to that molecule, you have to do it to prove it. So it is predictive in a way, and conclusively you have to do the research in the lab. That is the only challenge, and it is there with all, because AI we are feeding data and then it is doing the whole churning and giving us the result. If that information is not available, it won't be able to give us information, correct information.
Ma'am, one more question is there. The question is, can molecular insights help in developing a better targeted drug delivery system?
Yes, yes, it will, because the targets are known, and we know the through modeling and all, we know what is it, the pocket size and everything. We know what is the mechanism working on that particular target. If all that is there, it is very easy, and it is very fast also to understand the molecules, and to design the molecule also to help them hit the target.
Okay. Ma'am, one more question is from the B Farm student. The question is, what basic skills should a B Farm student develop if they want to work in the molecular or biological research?
So this is from BCom. This is from which B Pharma?
B Farm student, ma'am.
BCom.
B Pharmacy.
B Pharmacy. So
Yes.
B Pharmacy, see, pharmacy has an advantage. You know what? They have very good background data of the plants, the drugs, and their mechanism. The linking factor is, so this is BCom. When you clear your net and come to do research, you have the basic information in your mind that this particular compound or molecule or bioactive present in the plant, it will work this way, right? So if that idea is there, the knowledge or information is there, it is highly compared to other students, easy to understand which molecule you should work on. And that is the foremost qualification to do a research. So for a research, you need to know what is that you want to work on, and what all information is available for that particular molecule. And these two information is available, then it is only Google that tells you what all has been done, what is not being done, and how to work on it. So B Pharma is a very, very good platform to start your research when you, after clearing your NET exam. That is something which I will suggest to all the UG and PG students, that please do not waste your time in doing sideways projects. Either you stick to your passion, what you want to do, or you clear your NET and come to research field where you have 5 years to prove your research capabilities and with a fellowship.
So much, ma'am, for such an enlightening and insightful presentation. Your talk has highlighted how multi-omics technologies are transforming plant research and helping us in understanding the biological mechanisms at deeper level. So, Salish sir, now I request Salish sir to please present a formal vote of thanks. Sir, please.
Very good afternoon to all and one present in this meeting. I'm very thankful to Dr. Sulfa Gupta ma'am from their busy schedule who gave the time for our lovely and sweet IPGA Women Forum members to enlighten on a very specific topics of molecule to the mechanisms from multi-omics. So thank you, ma'am, first of all, to join us and give your precious time. And I'm thankful to Dr. Manju Singh, ma'am, Chairman IPGA Women Forum, Chairperson, Co-Chairperson Dr. Vandana Garg, madam, IPGA Punjab State Coordinator Dr. Neelam Sharma, Haryana State Coordinator IPGA Women Forum Dr. Geeta Deswal, ma'am, for organizing a wonderful seminar on the plant omics. मुझे लगता है कि आज के टाइम में हमारे स्टूडेंट्स कहीं ना कहीं प्लांट से और प्लांट की स्टडी से कतरा रहे हैं। हमारी एक ब्रांच फार्माकोग्नोसी, शायद उसमें भी कहीं एडमिशन नहीं हो रहे हैं या फिर उनसे बहुत दूर भाग रहे हैं। बट मैडम, आज का जो आपने टॉपिक सिलेक्ट किया और जो बच्चों को इनलाइटन किया, सारी स्टूडेंट्स को, टीचर्स को अपनी नई रिसर्च के साथ, इट इज़ वेरी वंडरफुल। और उसको देखने के बाद लगा कि एक अनचूहा पहलू भी एक रह गया है जिसे हमें जरूर टच करना चाहिए बीइंग अ फार्मासिस्ट। एंड हाँ, नेचर से ही हमें सब कुछ मिलता है, ये तय है। और यदि नेचर से हम और कुछ अच्छा ले सकते हैं तो हम नई मेडिसिन और उनका इलाज और अच्छे से कर सकते हैं। Last but not least, I am thankful to the Dr. Kumar Gaurav sir, who gave a very nice platform to all the delegates and to the organizers of the program. Especially thanks to Dr. Anjali madam, who
Thank you, sir.
really
My
do
pleasure.
a great work. And I am thankful to all the delegates also, जिन्होंने इस प्रोग्राम को सुना, देखा। और ये एक रिकॉर्डेड सेशन है, आगे भी जो चाहेंगे उसे और देख सकेंगे। So thank you very much for inviting me also in this part of this seminar. So thank you very much to all of you.
Thank you.
Jai
Thank you
Hind,
so
Jai
much,
Bharat.
sir. Thank you so much, sir, for presenting the vote of thanks. And thank you, ma'am, for enriching us with the knowledge and sparing your valuable time for giving this seminar. Once again, thank you all. Thank you.
Thank you so much, sir. Thank you so much,
Thank
ma'am.
you. Thank you, ji.